Hi everyone, let's talk about DNA sequencing. Now there are many ways to do DNA sequencing. The one that we're going to be talking about here is the Sanger sequencing method. So that is the method that this book focuses on. Before we talk about the Sanger sequencing method and what's the principle and how we do it, let us talk about sequencing in general. So the sequence of the DNA nucleotides of most organisms is unknown. We don't know the sequence of the nucleotides in DNA of most organisms. Now, knowing the sequence of an organism's DNA or of a cloned DNA fragment provides scientists with valuable information. What is this valuable information? It is important to know the benefit of DNA sequencing. So these are the benefits of DNA sequencing. First, you can predict the sequence of a gene. So the sequence of a gene can be used to predict the function of the gene. So once we have this sequence, we can predict the function, right? Second thing, to compare genes with similar sequences from other organisms. The third thing is to identify mutations or errors in the DNA sequence. Once you have that sequenced DNA, and you know that there's a mutation or an error, this can be helpful in the future, right? So these are the three things that DNA sequencing provides us with, these are the three valuable information that scientists get once they do DNA sequencing. All right, so because the genome of most organisms are made up of millions of nucleotides, the DNA molecules used for sequencing reactions must first be cut into smaller fragments using restriction enzymes. So when we're sequencing DNA, we're not looking at this whole huge genome. No, we're just taking a specific fragment of DNA and we cut that using restriction enzymes. Okay, so Sanger sequencing is based on something called dideoxynucleotides. Now, what are dideoxynucleotides? To understand that, we've got to look back at our nucleotide. Now, a nucleotide has your sugar, your 5-base sugar, your nitrogenous bases, so A, T, C, and G, and your phosphate group. Now, in this 5-base sugar, we've got the OH group. Now, when we get rid of one of these OH groups, and now we don't have it, it's called a deoxynucleotide. Now, this hydroxyl group right here on the 3' end plays a very important role in the DNA polymerizing reaction, and that basically is when we're sticking bases together. So what happens is in the DNA polymerase reaction, this OH group has a reaction with the phosphate group of another nucleotide, and that's what allows them to stick together. Now, if this OH group is absent, like we have in deoxynucleotides, then we won't be able to connect a nucleotide to this one because the OH group is absent. So let's rewind. On the 3' we have the OH group. Now this OH group is very important because in the polymerization reaction, what happens is this OH group has a reaction with the phosphate group. And that's what allows the nucleotides to stick together. But if this OH group is absent, we don't have it, that means no other nucleotides can stick to this nucleotide, right? It cannot connect with anyone else. It's like when you try to hold hands with your friends in a circle, but one of your friends doesn't put his hand into his shirt, so he doesn't have his hand out. So you can't go and grab his hand, so you can't connect with him, or he can't connect with anyone else because he doesn't have his hand to connect with someone else, right? So that's the same idea here. If we lose our OH, that means our sugar is losing its hand, right? It's tucking its hand away. It's not open and accepting. We don't have the OH, so we can't have a reaction between the OH and the phosphate group, which is what allows them to stick together. Okay, let's rewind to our nucleotide base here. So when we lose the OH on the 2' end, we call it a deoxynucleotide. When we lose the one on the 3' end as well, we call it a dideoxynucleotide. So dideoxynucleotides are the nucleotides that are used in Sanger sequencing. Now let's look at the classical way of doing Sanger sequencing. So our template DNA, our DNA fragment, is divided into 4 tubes or 4 Eppendorfs, okay? And each tube would contain each tube would have a primer, so a RNA primer. It would have DNTPs, which we said DNTPs are deoxynucleotides. We said that they don't have the hydroxyl group on the 2' end. So you'd have DNTPs of A, T, G, and C. And what that would look like, we'll see examples of how these are written. You'd have DNA polymerase. Obviously, in each tube, you'd also have, and this is the thing that's different, one radio labeled, which is, in the book it says, a small amount of each nucleotide is tagged with a different color fluorescent dye. So that's the one radio labeled dideoxy nucleotide. And we said dideoxy means that they don't have both of those hydroxyl groups. And most importantly, they don't have the one on the 3' end. What does that mean? That means the reaction is going to stop once we've connected to this radio-labeled nucleotide. Okay, so we have this in each tube. What happens now is you have your template DNA here, right? That's your template DNA. You've got your primer, and then in the tube you have your DNTPs, and we write them like this. So D, ATP, that means I have base A in it. D, TTP, for example. D, CTP, let's say. DD, ATP. Okay, so these guys with just one D are the deoxynucleotides, but the one with 2 Ds means dideoxynucleotide. So this is the one that has the fluorescent dye. This is the one that's radioactivated, right? Okay, so what happens is we've got our DNA polymerase and our primer and whatever, and we're just going to go through normal DNA, like attaching the nucleotides. So what's going to happen is you have your the ATP, and it'll come up here and it'll be an A, and then you've got T, and then C, for example, and then you have this dideoxynucleotide attached. This dideoxynucleotide, and it has a fluorescent dye, and it'll just show up differently, right? Here, the reaction would stop. We would stop adding nucleotides. Why? Why do we stop adding nucleotides? Because this DDATP or this DDNTP, this dideoxynucleotide, doesn't have the OH group on the 3', the hydroxyl group on the 3'. So that means we cannot connect it to another nucleotide because we can't have a reaction between the hydroxyl group and the phosphate group. So the reaction would stop and it would terminate. Now, this process keeps repeating on and on until we have something, and this reaction is totally random, until we have something that looks like this. Now, this reaction will happen multiple times in that Eppendorf or in that tube, and you'll end up with something like this. So what happens is we're going to put these into gel electrophoresis, and what gel electrophoresis does is it separates these fluorescent tagged fragments by length. So the common question is, all right, I got the fluorescent, it's sticking up saying A, this is my color, see me, but how do I write the sequence? How do I find out the sequence? Well, you've got to listen carefully here so that you don't get lost. So we know that gel electrophoresis separates our DNA fragments according to size. So once we put, for example, let's take the first fragment here, into gel electrophoresis, it'll say you have 5 base pairs. I'll just write it as BB, sorry. So you have 5 base pairs, right? 5 base pairs. Okay, what does that tell me? How does that help me? Well, we know that this fluorescent dye radioactive nucleotide is a dideoxynucleotide. That means it doesn't have the 3' end. That means it cannot connect to any friends. That means if I say I have 5 base pairs, this fragment is 5 base pairs long. That means at the 5th base, the reaction stopped. We don't have any more bases. Why don't we have any more bases? Oh, because we have the radioactive A. What does that tell me? That tells me that there's an A on base 5. How did I know that? Gel electrophoresis told me I have 5 base pairs on this fragment of DNA. I know that this DNA fragment is 5 base pairs long. I know that I put radioactive DDATPs in there, right? And I know that they cannot connect to anything else. They're friendless. They ain't got no connection hand to connect with other nucleotides. They lost that OH group, that hydroxyl group. So I know if a DNA fragment is 5 base pairs, that means the last base pair is base pair number 5, and I know that we stopped at base pair number 5 because we have that radioactive A. So now once I write my sequence, I can go, okay, base pair 1, base pair 2, base pair 3, base pair 5, I've got an A. And this just keeps on happening in all the 4 tubes simultaneously, and you put it into this system, and an automated sequencing machine prints out the sequence. Now, if we just look here at what the book has shown us here, right here. So we put our DNA fragment into the Eppendorfs, as I said. So in G, we're going to have a radioactive G. In AA, we're going to have a fluorescent or radioactive A. In tube T, we're going to have a radioactive or fluorescent T, and so on. Then we put them into gel electrophoresis, and we separate them by length, right? And then they go into this machine that's going to write out the sequence. Now, how does this machine know that we have a G or we have a T or we have an A? Since, for example, let's say this is, since it's at the top, it's the largest, let's say that is 300 base pairs, all right? And it has, and it has the color purple, right? And we know that we put purple for G. So I know that at the 300th base, there's a G because the reaction stopped there. It would only stop there if something terminates the reaction. What terminates the reaction? The radioactive G. So if my fragment is 300 base pairs long, that means that this radioactive G is at base pair number 300. So I can say I have a G at base pair 300, right? And this happens in all of those 4 Eppendorfs, or in all of those 4 tubes simultaneously at the same time, until you end up with the DNA sequence written out for you. Obviously, this can't be done with a human. You need an automated sequencing machine to do this. But that is the idea behind DNA sequencing. So, as we said, the sequence of the original DNA template is determined from the order of the tagged fragments, or the radioactive fragments, or the fluorescent fragments. And we can read that using gel electrophoresis, and that is the way that I explained before, looking at the length, seeing where the base pair is, how many base pairs we have, and then writing out the base pairs, and that is done by system.